Okay, welcome everyone for joining us for the study seminars this quarter. And today it's our great pleasure to have Eardi, Lila from Biostatistics Department. Eardi joined the UW at, I think, early last year, right? And then Eardi graduated from University of Cambridge, working with John Aston. And today he's going to talk about functional random effect modeling of brain shape and connectivity. So let's welcome Eardi.
Yeah, thank you. Thank you for the invitation, Yinchi. It's my pleasure to be here today. And by the way, you can stop me anytime to ask questions, either by putting them in the Q&A chat or just asking them yourself, really. So today's talk is going to be fairly applied. And I mean, the emphasis is going to be the analysis of sort of brain imaging data. Okay, so I was like, why not start with a brain image, right? And so this is a reconstruction of a human brain. So you have to think of this as a sort of brain that is inside the inside the head of a person who is looking at your right shoulder, basically, right? So that's how you understand the orientation of this brain. And you might agree that there's a lot of information in there, and there's information both of structural type, so sort of description of the anatomy of this brain, right? And the functional type, functional in a sense of, in a sort of neuroscience sense, which means a sort of description of how the brain works, okay? And but let's start from the structural bit of it. So you can see that there is this sort of two-dimensional surface embedded in this three-dimensional space, and that surface describes the geometry of the brain. And really, really what I mean by geometry of the brain is the geometry of the outermost layer of the brain, okay? And you might wonder why we work only with the outermost layer of the brain, and that's because, I mean, that's where the sort of neuronal cell bodies are. So roughly speaking, that's where the thinking happens, on the surface of this outermost thin layer of the brain, and not on the inside, okay? In case you haven't worked with this data, you haven't seen this data before, what's on the inside of the brain, that's mostly wiring, right? So axons linking the different parts of the cortical surface. So you might agree with me that this surface here is a description of the geometry of the brain, so of the anatomy of the brain, right? But of course, what makes the brain interesting is not just the anatomy, but also the way the brain functions. And this map here, the color map that you can see here, is instead a description of how the brain functions. So imagine green values are low values and red values are large values. Then we can, this signal here represents, or that number really represents how hard every single part of this brain is working, okay? So if you see a red area, it really means that that area is working a lot, okay? If you see a greenish area, it means like that area of the brain is working a bit less. So it's not surprising that this color map is only on the brain surface, only on the cortical surface, how we call it, because that's where I said the thinking happens. That's where the relevant signal comes from, right? And what else? So yeah, this really describes how the brain, how hard every single part of the brain is working at a specific time, of course, right? But what makes the brain interesting really is the fact that we react differently to different situations. We react differently after we've had certain experiences. So really the fact that the brain from a function point of view is a time varying object, right? So it's not surprising that this is only an approximate description of how the brain functions, or better, this is just a snapshot in time, okay? But really we can collect this signal related to brain activation over time, okay? So really this signal here is a time variant signal, okay? That changes over time according to, for instance, what the subject is doing, okay? In this specific case, this is a real signal, by the way. We'll see later that this comes from fMRI, so comes from a sort of MRI machine, and is describing the brain function over time of this specific subject, okay? And so there are these two elements here, brain structure and brain function. And by the way, just to put things in perspective, I mean, when I talk about how hard one part of the brain is working, if you take 1 millimeter cube of this brain here, of this circular cortical surface, that's more or less, I say, multiple 100,000 neurons, right? It's just a very rough description at a sort of neuronal level of what's going on, right? So there are two important elements here. We said the structure of the brain and the function of the brain as a time-varying signal, okay? So you'll see that during the talk, we will put a lot of emphasis on the sort of geometry of this data, okay? And geometry comes in at least two ways, okay? So the first way geometry comes in is that you can see that this is sort of a non-linear surface, and you can see that this signal here is sitting on top of this non-linear surface. So when you propose statistical methodology to analyze those signals, for instance, you want to account the fact that this signal is sitting on top of a nonlinear geometry. So the way, for instance, information propagates, or the way we learn, for instance, in a non-parametric way about one point at looking at the neighborhood, the concept of neighborhood is a bit different here. It's constrained on this surface, okay? But that won't be the main type of geometry we will focus today, okay? We will look at another type of geometry, a bit more abstract, okay? And that comes in really if you look not only at one surface, okay, but if you look at multiple brains. So suppose the same exact image, okay, type of image, but for multiple subjects, okay? And why there is a geometry if you look at multiple subjects? Well, because we know that brains are likely to be shaped in a certain way, okay? They are less likely to be shaped in another way, but we are 100% sure that the brains are not shaped in some specific way. For instance, we know that there's no such brain as a self-intersecting brain, so where this surface comes out, self-intersects itself, and then comes out again, right? That's a surface that you can get by playing with these mathematical objects, but not a brain you can get, right? So this adds some constraints on the space of possible brains, and that's the type of geometry we'll mainly be talking about today, okay? So by the aim of this talk really is like, what if I get to observe 1,000 examples of this brain, right? And each of these brain, as we said, is a pair of information, both anatomical information and functional information. So if I get to see 1,000 of these examples, can I learn something more general about, I don't know, how specifically shaped brain work, or how brain that work in a specific way, so they have a specific signal, how those brains are shaped, okay? So that would be the aim, really, have 1,000 of these brains and learn something about that generalizes well from these 1,000 examples. So as I said, there is a lot of geometry in this data, and not because we like geometry. I mean, we do like geometry, but also because that's the relevant things to do from a sort of neuroscience point of view, right? So these geometric data come naturally in this setting, okay? And you might wonder at this point, where do these data really come? How can I actually acquire this data, right? Sort of the engineering process of it. And turns out that those geometric data really are just MRI data that have been pre-processed to have that form, okay? So maybe you're a bit more familiar with MRI data, but as you know, MRI data are grayscale data, okay? And they are very plain. Okay, so they are flat. Okay, here, for instance, on the left-hand side, you see one section of an MRI. Okay, and this section of an MRI gives a description of the structure, of the internal structure of the brain, as you can see, right? And of course, we don't have only one slice, but we have many of these slices. It's really like a 3D image, okay, that describes the internal structure of the brain. Now, if you remember what I told you about the neuronal cell body, so the important bit being on the outermost layer of the brain, you can see here that you can distinguish two types of tissues. The dark gray one, which is on the external bit, okay, that I'm trying to follow with my cursor, and the internal bit, which is called white matter. And as I said, the important bit here really for the type of analysis we want to do is this external layer that we can see here, okay? And what happens is, given that we care only about that layer, what happens is that you can actually segment, is a typical preprocessing step, you can segment out only the outermost layer of the brain and fit a surface to it, okay? And that's how we get the cortical surface, okay? So this is how we actually managed to get this image, which is exactly basically the brain shape, the image that I showed before. And then at this point, you might wonder, well, what about the brain function? So how do I get the brain function information? And it turns out that that's same machine, the MRI, but a different modality called functional MRI. So functional MRI captures a sort of 3D signal, so related to brain activation, okay, a bit like the MRI. But this signal, rather than describe the internal structure of the brain, this signal describes the brain activation, okay? And really captures this 3D image over time. So it's really a 4-dimensional image, okay? But as I said, the important signal is really the one that is on the cortical surface or the outermost level layer here. So what people do typically is that they go on the fMRI, they select the location that are important, those that are on the outermost layer of the brain, and extract those, okay? So if you fix one location, you will have a time variant signal that describes brain activation at that specific time point that you see here, okay? So how can we put everything together? Well, now we can take this signal here, okay, and map it onto the surface. So take only the signal that is relevant, so on the cortical surface, and map it into this surface, okay? And the final type of data that we get is this structure here, this item here, okay? Which is exactly the same type of data that we had at the beginning, just I don't use the video to display them, but I use this time series to display them, in the sense that we have, again, a surface that describes the brain geometry. And in this case, I use time series to represent them, but if you fix one point on this surface, you have also a time series describing how that specific part of the brain is activating over time. Okay. And this is really this would be, which is a bit strange, this would be one statistical sample of our analysis, right? And we suppose we can do this for multiple brains, and that's where we'll really start doing statistics. So our most atomical element of our statistical analysis is really this pair of objects, the structure of the brain and the function of the brain. Okay. So I hope I sort of convey the idea that this geometric structure, this geometric data really sort of pop up naturally in this sort of neuroscience setting. So now we'll move on and see what are the sort of the aim of our statistical analysis here. So here in the central panel, okay, you can see I display again the same data that I showed before. So one subject, description of the anatomy of the subject, and description of the brain function of that subject. Okay, so how every single part of the brain is activating over time. So how do we go about analyzing this data? Okay, first of all, in this specific experiment, the resting state, this fMRI signal that we can see here, this related to brain activation, was acquired while the subject was not doing anything specific, okay, while the subject was not performing any task. And that's why they are called resting state data, okay. So it's not really important to look at the signal, whether the signal goes up or whether the signal goes down in every single point, because fundamentally that will depend on what the subject is thinking at a specific time, for instance, right? But what's important in thinking in terms of connectivity, so how the different parts of the brain work together, okay? And the way we construct an object, and this is very typical in the field, the way people look at connectivity when it comes to brain is that they pick 2 points on the brain. So suppose this point here and 2 points here, this other point here. And then you can, of course, extract one time series for every one of these 2 points. Now, if you want to understand how well together those points are working, as opposed to whether they're working independently or uncorrelatedly, then you can compute the correlation between these 2 time series, okay? And for every 2 points, that will tell you how well together those 2 points in the brain they're working, okay? And of course, you don't want to specialize your analysis as only 2 points. So what you do is that you do this for every possible pair of points, okay? And you put them in this, you organize this correlation in a sort of correlation matrix, okay? Where every row indicates the position of a first point on this brain, every column denotes the position of a second point on this brain, and the element in that specific row and that specific column will tell you something about how well together those two parts of the brain are working. Okay, so something about how connected those two parts of the brain are from a functional perspective. So really, when you're performing it, so really in terms of brain function. Okay, so this object here, you can imagine, is high dimensional. It will probably be too high dimensional. So rather than do this for every single pair of points, people focus on parcellations of the brain. So parcellate the brain in different areas, like these colored areas of the brain here, and really do this, compute this connectivity matrix only for the sets of your parcellation. Okay, and that makes the dimensionality of this object a bit lower. And this object here is called sort of connectivity fingerprints, as I said, because it's a fingerprint of how the brain of that specific subject is connected. Okay, and this is a description of the brain function, of functional connectivity. Then, of course, we have the surface here. So what we do is get rid of the signal, okay, and just keep the surface, and that will be a pure description of the anatomy of this brain, okay, or in other words, of the brain structure, okay. So really, the two fundamental elements in our analysis will be this object here on the left, describing brain structure, and this object here on the right, describing brain function. But as I said, this is really for one subject, subject one. And I can, of course, do the same exact thing for multiple subjects, so n subject, okay, where for every single subject, I have a description of their anatomy of the brain and their connectivity of the brain. And as I said, my aim here would really be, can we, from this set of 1,000 examples, and here will be 1,000 in our final analysis, can we, from this set of 1,000 examples, learn something more general about the relationship between shape and connectivity, okay, of the brain? So relation about structure of the brain and connectivity of the brain. And this is, by the way, important, right, in practical settings, because we know, for instance, that in the case of neurodegenerative diseases, that sort of change the anatomy of the brain due to the disease, we know that that changes the brain function as well. For instance, we know there is a phenomenon called plasticity, where different parts of the brain take over the task of the brain, of the part of the brain that is missing due to the disease, for instance, okay? But there are also other situations where we know that performing specific tasks over and over, so people with specific connectivity fingerprints might lead to changes, maybe not these macroscopic changes, but might lead to changes in structure of the brain, okay? And there is one more element in terms of data, and it's the fact that these 1,000 observations are not 1,000 independent observations, okay, because some of these subjects are related. So it turns out some of these subjects are monozygotic twins, some of these subjects are siblings, some of them are half sibling, and some of them are independent pairs, okay? So there is a kinship, what is known as a kinship structure across the n elements, okay, that is basically a description of the family structure. And this is known, okay? We exactly know which one of the two subjects have this, what are the two subjects that what are the subjects that are identical twins, what are the subjects that are siblings, and so on, okay? And what are the subjects that are independent? So this type of structure, non-independence across the samples, is somewhat annoying, right, in principle, because doesn't allow, you would rather have them independent, right? Because it doesn't allow really to span all the space as well as the sort of, as well as this observation would do if they were independent, right? But in actual fact, we will try to take advantage of this family structure to try to disentangle sort of variation that is due to genetic factor, okay, variation that is compatible with this similarity structure across sample, and variation that is non-compatible with this kinship structure, so it's more like independent. So kind of variation that is due to environmental sources as opposed to genetic sources. So really the final aim will be like understand relationship between shape and connectivity that is due to genetic sources, and relation between connectivity and shape that is due to environmental sources. Okay, so next step will really be, okay, so what are the challenges here? Challenge is that one statistical sample here is an object, and actually is a pair of objects. It's not a typical number that you get in bivariate analysis or the typical vector that you get in a sort of multivariate analysis, okay? And the other challenge is the structure across basically the rows of your data matrix, as a applied statistician would say, which is the structure across your samples, okay? So we will first see how we deal with the structure across the samples, so with this kinship family structure, and then we'll see how we deal with this with the fact that our observations are objects, really. So let's start simple, just to make the point of how we model family relatedness. And suppose now for a second that shape is not this complicated object, but shape can be summarized with one number. Okay, suppose the volume inside the surface. Okay, suppose someone told us everything you need to know about the shape of a person is the volume inside this surface. Okay, so you have one number for every subject describing one number for every subject describing their shape. Okay, and suppose you can do the same exact thing for connectivity. So you have suppose you can summarize this big matrix with one number, which is, say, a number of total connectivity. And you can do this for every subject. So for every subject, you will have only one number describing their connectivity. What happens is that you can take that for every subject, for every rows of this matrix here, you can take the one number that describes shape and put it next to the one number that describes connectivity, and do this for every single subject, right? And then what you can do is put together a basically n times 2 matrix, which is your data matrix, where the first column describes shapes, one number, and the second column describes connectivity, a second number. Now, of course, this won't be our final setting, it will be a bit more complicated, but just to make the point, because this is just enough to see how we will model structure across the rows of this matrix, or structure across the subject of this analysis. And the way we'll do it is we will think of this joint data matrix A, where there is both shape and connectivity, as given by the sum of two contributions, G and E. Okay, now, this is a bit crazy, right? Because how can you hope to identify the sum of the two matrices whose sum give you another matrix A, right? And the idea is that maybe we can in this setting, and it's not really our idea, that's the idea of the variance component model, bivariate variance component model. The idea is that maybe you can, if you assume something about the distribution of these two contributions, which are matrices n times 2 and another matrix n times 2, something about the distribution of these two matrices. And we assume that they are matrices, they're basically normal matrices, okay, that are described by 3 parameters, where the first parameter is the mean. Suppose we have removed the mean, so we have no mean. The second parameter is a correlation structure, a covariance structure between the rows of the data matrix, which is the new element, the one we usually don't have in a normal statistical analysis. So this would normally be IN if the samples were independent. And then a third element, which is the covariance between the columns of this data matrix, which is the important thing, understand something about the relationship between shape and connectivity. And as you see, the genetic, G stands for genetics here, the genetic contribution, we assume that there is structure across the rows of this data matrix, and this structure is compatible with this matrix of relatedness coefficient that describes the family structure of our data. And it's given, so Kn is given here, as opposed to the environmental contribution, which is instead compatible with an identity matrix here, which means an independent structure across the rows of our data matrix. So what are the elements to be estimated here are sigma G and sigma E. I would like to estimate the covariance structure between these two columns that is due to genetic factor, which means it's compatible with this family relatedness structure, and the covariance structure across the columns of this A that is compatible with an independent structure across the rows of my matrix. So this is a classical. So in the end, I get this sigma G, which is a 2 times 2 matrix in this oversimplified framework, which basically describes the variance of shape that is due to genetics, the variance of connectivity that is due to genetic, and the correlation between connectivity and shape that is due to genetics. I get another matrix sigma A, which is the same exact thing, only about connectivity. Okay? And the way we estimate this is a maximum likelihood approach. REM really is restricted maximum likelihood. So just write the maximum likelihood of this object, and you minimize over sigma G and sigma E as the elements minimize the likelihood of this object. You will wonder, what does this have to do with the with a complicated setting of having a shape that is not a number, but really an object, and having a connectivity, which is also this complex object? Well, we'll see that we'll be able to translate the problem that we had before, maybe not in a bivariate setting, so maybe not just one number describing shape and not one number describing connectivity, but we'll be able to translate the problem, the difficult problem, into a problem where we maybe have, I don't know, P1 numbers describing shapes and P1 numbers describing connectivity. And then you can see how this model can be generalized to the setting where you have multiple data describing shape and multiple numbers, so multivariate setting describing connectivity. So this is how we really deal with the structure across the columns. And then we'll get back to this once we see how to deal with the traits really being connectivity and brain shape. Okay, these are complex traits. So we have somewhat to summarize them in with some practical quantities. So what is the practitioner's approach? The practitioner's approach is that the practitioner sees this shape, okay, all this complicated shape, and really the practitioner would like to translate this shape into a data matrix, right? Because once it's able to translate this shape into every single shape into a row of your data matrix, then can take the data matrix, launch the favorite R function on it, or Python function, and basically have the analysis done, right? So what are typical features that one can extract? Well, enclosed volume, as I mentioned before, total surface, average curvature, and so on. Okay, so you can come up with P features, okay, extract the P features from every one of the brain and put them, fill every single row of this data matrix. This is not what we do, though, because there is an issue with this, which is a bit more abstractly maybe. This map, the map brain to features, a bit like a bit like the word to vectors type of things. In this case, we have a sort of shape to vector type of mapping, is non-invertible, which means once you have these features, you've lost information and you are not able to go back and see what is a brain associated with, what is the brain that is associated with those features, right? So suppose you have this data matrix, you run your favorite machine learning model, you learn something about the columns of this data matrix, which means about the features, okay? But you cannot translate the results back in the space of these original objects because this map is not invertible. So we will rather take a different approach, which is a bit the non-parametric approach. The non-parametric approach consists of taking these points here, where we call landmarks. Okay, so taking many of those. Here I display only 6 of them, but suppose you have 64,000 of them. So basically enough to fully describe the shape of the subject. And once you fix these landmarks, find out where that exact location of the brain, suppose I'm working with this point here, the point J, find another landmark in every other brain that is in the same exact position as that brain. So they are in correspondence, right? As I said, suppose you have many of these landmarks, you solve the first problem, which is you have a full description of the brain of a specific subject. Then what you can do, for instance, is that if you take for every landmark, suppose this is landmark 1, if you take the coordinate, the space coordinate of that landmark for subject 1, and you put them in a data matrix and fill the first 3 columns, and do the same for the same landmark for the other subject and fill the first 3 columns of the other rows, then you have basically now we've solved one of the problems, which is this data matrix offers a better description, basically almost a non-parametric description of the geometry of the brain here, right? In terms of a very dense set of landmarks. I have no way to see what, oh yeah, time. Okay, so we've solved the first problem, I said, which is give a sort of better description of these geometries here. And by the way, the price you're paying already is that this matrix P in this matrix is huge now. So you can think of, if I have 64,000 landmarks here, times 3, that will be in the order of 200,000, okay, as opposed to n, which is in the order of 1,000 in our analysis. So is it done? Can we really just do statistics on this data matrix here? Well, as I said, there is some geometry in there, but we are not yet modeling. And the point I'm making now is that suppose you're doing your classical multivariate analysis and you're working with a data matrix X, which is n times p. Okay, suppose feature 1 is something like height, feature 2 is something else. Anyway, this is your classical analysis. One way of displaying your data, if p is small, say 2, is to scatter plot them, right? So you plot the coordinates of every single row of this data matrix. And this is what's something that you will typically get, right? Why I'm saying this, because if you were to do the same, and this is really abstractly, right? If you were to do the same with those features that you have extracted from the brain, so those locations of landmarks, and you were to plot them in this 200,000 dimensional space, we claim that you would not fill the entire space, but you will fill only a subspace of your 200,000 dimensional space. Why? Because as I said before, there are configuration of these landmarks that are plausible, like the one we see here, but there are also configuration of these landmarks that are not plausible, things like self-intersecting shapes. So really, your surfaces, as I try to sort of pictorially represent in this image, lie in a subspace of your 200-dimensional space. So what's inside this space? These are valid shapes. Forget about this tangent space for a second. These are valid shapes. What's outside this space is shapes that we don't see in practice, so are shapes that are not physiological, that don't have physiological configurations. So why I care about modeling this subspace, so this geometrical subspace where brains live? Well, because if I want to do prediction, for instance, I would like to do prediction with brains that are physiologically valid brains. So not predict something that lives outside this space. It's really like a logistic regression, as I like to say, where you constrain your output to be a probability, so something from 0 to 1. Here, you would like to constrain your prediction to lie on this low dimensional subspace that we'll see have the sort of mathematical structure of a manifold, okay? And that's what we are trying to do. So the additional challenge in working with this data is that this data live in a subspace of your 200,000 dimensional space. So how do you do statistics on these subspaces that are manifolds, let me say? Well, you have to redefine both the population object and the sample and the empirical object of every single thing you know, basically, starting very much from the mean, right? So you have to redefine the mean. Why? Because if you were to take the simple average of these points, as you see, the simple average might fall outside the geometrical constraints that you have. So it might be a non-valid shape. So how do you define instead the mean? Well, you have to add some structure to this geometry. You have to define a geodesic distance, which is a distance that is measured while you are constrained to stay in this space. So the distance between xn here and x1 is not the straight line. The geodesic distance is not the straight line, but it's the line that follows the surface. And once you define a geodesic distance that I call dg, you can measure the distance in a sensible way between pairs of objects. And once you can measure the distance between a pairs of objects, it's easy to generalize the concept of the mean as the barycenter of this object. Basically, it's the object x that is constrained to this manifold, to this geometric structure, and that achieves the aim of being the points that is that is not far away from everyone. So minimizes the distance from all the points that you observe. And this is a generalization of the mean to non-Euclidean spaces or to manifolds, which really means non-linear manifolds, which really means spaces that have some geometry, which is not the elementary geometry of R3, where you can draw straight lines. And then to really do statistics on this object, you need a bit more structure. You need a Riemannian structure, it's called. So Riemannian structure, what we really care about here is the fact that it comes with some canonical maps. And these canonical maps are the Riemannian exponential and Riemannian logarithm. So Riemannian logarithm is something that takes you from this curved space, so it takes a brane from this curved space to a tangent space representation. Okay, so we can represent this surface here that is in a nonlinear space, Xn, with an object that is in the tangent space, which is linear. Okay, and this canonical map that is called logarithm or Riemannian logarithm is the map that takes you from here to here. So the idea would really be, what if I map all my object in the tangent space with a logarithmic map, which is a linear space? I do linear statistics on it, for instance, principal component or functional principal component in this case. And then once I have the results on my tangent space, I can use the exponential map to map them back into the nonlinear geometry. Now that's exactly what we do. We estimate these tangent elements here, and we do principal components on these tangent elements, which will give us a set of features, say PS features, PS equal to 10, for instance, for every subject, for every one of the n subject that describes the shape, the brain shape of that subject. So we don't have a lot of time, but just briefly, this is of course a sort of pictorial representation of what's going on. In practice, these objects are sort of functions, right? So I won't go into the details of this, but the idea is that the elements of these tangent spaces are really smooth vector fields in R3, like the vector field that you see here. And the smooth spaces, Hilbert spaces, are linear spaces. You take linear combination of those, even extrapolation of those, and those will always belong to the initial vector space, right? And we use this linear space, which is a space of smooth vector field, to generate diffeomorphic deformation. Diffeomorphic deformations belong to a nonlinear space, but the nice thing about diffeomorphic deformations is that they preserve the structure of the brain. So you will never get self-intersecting brain. So in practice, what happens, forget a bit about the math here, but what happens is that we can, with an element of tangent space, or a tangent space, like this one here, this vector field, we can always represent, by solving a specific ordinary differential equation, an element of the nonlinear space, which is this deformation here at time 1. So now, okay, which is a representation of the brain shape, which instead belongs to a nonlinear space. So it's in this curved space. On the left-hand side here, we have basically one of these tangent space elements. On the right-hand side, we have an element of the diffeomorphic space, so nonlinear space, and we use smooth spaces to parameterize nonlinear spaces. And basically, it means that you can do PCA on these vector fields. So basically, in practice, is in RKHS, and which is a linear space. Okay. Then, as I said, this is the forward mapping. So given a vector field on the tangent space, you can always compute the exponential map, basically, and that gives you the element in the nonlinear space. Okay, but really the statistical problem is the opposite problem. So how do I estimate a vector field representation from a shape? And you can do that basically by using minimizing a penalized functional, where you're minimizing the norm of the vector field you're estimating, plus a measure of the distance between the deformed shape and the actual shape they're trying to represent. Anyway, this is perhaps the most important thing, which is linear parameterization of your nonlinear space. You might wonder, what about connectivities? Well, those are correlation matrices or covariance matrices, so they are constrained to be positive semi-definite. So that space also has a geometry, and you can it's a constrained space, and you can enrich it with a Riemannian structure, and also come up with canonical exponential maps and logarithm, and really apply the same idea. So have this object belonging to the space of positive semi-definite object, which is nonlinear, map them into a tangent space, which is linear, do statistical and tangent space, and basically compute these coefficients on the tangent space that give a compact representation of these objects. So, okay, there are many more details on how we define a distance on the space of the covariance, and that's pretty different from how we did in the space of shapes, okay? But we don't need to go into it. The general picture is the same. We find tangent space representations of these nonlinear objects. Remember, the aim here is really what we have achieved is that now that we have this tangent representation, we can compute a reduced representation of them and have a few coefficients, PC, that for every subject, they describe the connectivity of that subject, okay? And for shape, we did the same. We had few coefficients, say PS, that describe the shape of that subject. So now really we can go back to our variance component model, with the only difference that we have these PCA scores here that describe shape computed with this by doing tangent space mapping, okay, and functional principal component. And then you have these connectivity descriptors, which you have computed the same way. You have, say, 10 of them, okay? So the only difference from before is that you don't have one number for shape and one number for connectivity, but you have 10 numbers, say, from shape and 10 numbers for connectivity. So how do we account for the structure across the rows now? Well, with a variance component model. So you put them in a matrix A next to each other, which is n times 20, say, so 10 descriptors of shape and 10 descriptors of connectivity, and you try to separate the geometric contribution and the environmental contribution. And you get some estimates of the correlation structure between due to geometric structure and due to environmental source, due to genetic sources and environmental sources. The only difference is this matrix is not 2 times 2 now, because it's not a bivariate, not bivariate data, but it is, say, 20 times 20. So this correlation matrix is really describing the correlation between every descriptor of shape and connectivity with every other descriptor of shape and connectivity. Remember, it's correlation structure across the columns of this matrix. So we can actually plot these objects and you get this, basically. So as I said, you have to think of the rows and columns of this matrix as being the first 10 here are descriptors of shape, and the second 10 are descriptors of connectivity, okay? And this tells you how every descriptor is correlated with the other descriptor. If I were to take the simple correlation compute my simple correlation of my data matrix, of this data matrix. But of course, that's not what we want. We want the separation of this object into the sum of virtually the sum of two other objects, which is the correlation due to genetics and the correlation due to environment. And we can see interesting stuff here. I said the first 10 here are shape descriptors. So it seems like shape descriptors have variance 1 here, every single one of them, because they are PCA scores, so they'd be normalized. But if you look at how they are distributed between genetics and environment, it looks like there's much more into the environment side of it. And it's not surprising. It means that brain shape is somewhat more dependent on genetic factor than environmental factors, okay? As opposed to connectivity, which is this other block here, where the variance of every single descriptor of connectivity is more on the genetic side. It's 50-50 more or less. So it's equally distributed between genetics and environment. And it makes sense that the way we think, so the way we function, is modeled also by the environment rather than genetics only. And we repeat the analysis with and without confounders. It doesn't make a huge difference, at least at this scale. So, but as I promised you, these are not just descriptors of shape and connectivity, but are descriptors that we can map back into the space of shape and into the space of connectivity, thanks to those exponential Riemannian maps, okay? So in actual fact, we can take this connectivity structure and look at what are the most correlated changes in shape and the most correlate to the changes in connectivity that are due to genetics and environment, okay? I won't get into the exact description of this plot, but the idea is that here you can see changes in shape across subject. And what are the most correlated changes in connectivity to these changes in shape that are due to genetic sources? And we can do the same thing for the environment on this other side here. Okay. And you can appreciate some microscopic changes in shape. As I said, I won't go into the details of the descriptor, and some microscopic changes in connectivity. So it seems like the brain that look like more like this one here are a bit less connected, which means light red colors. And the brains that look like more like this one tends to be a bit more connected overall. But of course, you can study the single pairs of connection. Well, they are overall more connected. This looks a bit more red. And remember, we took this sort of non-parametric approach to an extent, which means there are not only global variation that we can capture in shape changes, but there are also local variation. And this is, I think, an example of an interesting local type of variation that we can capture. This is the back of the brain, so the visual cortex. And it seems like that people that have a specific connectivity tend to have a brain shape more like this one here in the visual cortex, and people that have the other type of connectivity patterns we saw in the previous image tend to have brains shaped more like this thing on the right. So the overall analysis looks like this one, as we said. We basically take this geometric approach to analysis of this data, so map them into tangent spaces, both shape and connectivity, disentangle variation that is due to genetics and environment, and we are able, once we learn something about these things, to map them back in the original space and look at trajectories in shape, so nonlinear trajectories of variation in shape and nonlinear trajectories of variation in connectivity that are most correlated, okay? So whose scores are most correlated. And I think this is what I have planned for today. So this work is in an archive paper. This is joint work with John Aston, who was my PhD advisor. And yeah, thank you everybody for the attention.
Thanks, Eardi. It's a very amazing talk. Yeah, and so actually, I myself have a questions about. Yeah, so I'm just curious. Like you mentioned that you can project it onto the tangent space and evaluate at the mean, right? So I'm curious. In this case, how do you estimate the tangent space? Do you use the fact that observations, because it's like you can actually just literally just for that specific point, just compute like using values of other observation and then kind of locally approximate that tangent space?
How you compute where the tangent space is centered, you mean?
Yes. Yeah. How do you obtain that tangent
space? So there
are
two approaches. Mm-hmm. One is you try to minimize this. You really try to minimize this objective function here. Mm-hmm. Okay. So you really try to come up with a C parameter. For instance, this is a covariance matrix C, mm-hmm. and that minimizes, say, gradient descent, minimizes an objective function of this type. Another solution is that you start with a tangent space centered at a random point. Mm-hmm. And you map your data with a logarithmic map on the tangent space. And if you see that the mean of your data is not 0, then you move in the direction of the mean, because there is this property that says that if you are minimizing this objective function, then the mean of your logarithm, the classical mean of your logarithms, because those are on the tangent space, should be 0. So those should be 0-centered random function, random variables. So that's another trick that people use. Project them and move in the direction where the mean is until you get a 0 mean.
I see. Okay, that's very smart. Thanks. And I saw a question from Nick. And do you need to standardize the shape data in a certain way to get meaningful estimates?
Yeah, that's a very good point. So yes, exactly, because we work with coordinates, with 3D coordinates. So if I were to play the exercise of taking a shape and randomly shifting it in a random location, and I were to try to compare a brain that is northeast with a brain that is here, that wouldn't work, right? In terms of coordinates are not comparable. So you really need to apply a sort of normalization, as you said, which more technically is called Procrustes analysis, which is really sort of a rigid alignment of these shapes to bring them as close as possible, okay, without changing their shape. And yeah, once you do that, then you can work with these coordinates. So yeah, that's a very good point.
Thanks, Eardi. And there's another question coming from Sam. And Sam says, thanks, I like your talk. And I was wondering if you could speak more on the trade-off of estimating the tangent space explicitly versus relying on the data lying asymptotically in the tangent space without making the logarithmic map explicitly.
Oh, that's a good point, but depends what you. I don't think there is any concept of there's not necessarily a concept of data lying asymptotically in tangent space here. Just to make the point, for instance, I think this is a good image here. Okay. You have to change also the population definition of the mean, right? Even the population definition of the mean. So if you had an infinite number of these brains on this nonlinear geometry here, and you knew the distribution of these brains in this nonlinear space, nevertheless, if you take the expected value, which is the population quantity, so the asymptotic quantity, that would nevertheless live outside the geometry that you have, right? No matter how many points you observe here, if they are around this point, the mean, even if it's the population quantity, would go outside the geometry where you want to constrain things. So asymptotically, things don't really work, as in fact, here I focus on the population quantity, which is the empirical mean, but you would have to redefine also the population mean. And the way you define the population mean is you basically keep this notation. You basically, again, just try to minimize this quantity, but just with the expected value of xi becoming x, but you change the definition of mean to be compatible with the structure. So there's not that much that asymptotics can help in terms of constraining your results to this valid geometry, I believe. Maybe sort of somewhat related to what you said is perhaps the concept of projecting. So what if I take a approach where I compute the mean classically, so I leave outside, and once I have a good estimate of the mean, I projected it on the subspace. And that's true. That's called the extrinsic approach, as opposed to our approach, which is called intrinsic. And that's computationally more efficient, but has the reputation of being a bit less robust to what we're doing.
Okay, thanks. And other questions? Oh, just out of curious, like I'm just curious, how do you choose the landmark points in the first place? Because I think I can see that if you choose that in a nice way, and I can see you need to have some constraint that they cannot be too close with each other, right? And that kind of. So I'm just curious, like how do you actually practically choose that?
I mean, that's a big topic in neuroimaging. So it's not really just us doing that. It's really they call the field of registration. You might have heard the topic of registration, and that's a very difficult problem. And the way it's dealt with, it's not even every sort of population analysis of this data needs the registration. Okay, so everyone works on registered data basically. But the main idea that is applied usually to register this data is that you inflate these surfaces until they become a sphere. And you do this for every single subject. So you map them to a sphere, which means parameterize them to a sphere, and then you rotate them, not just rotate them, but you do both linear and nonlinear registration on the sphere to maximize a concept of alignment, which could be alignment of the sulci, okay, or the functional data that are on top of it. So yeah, but it is a very difficult task, and we heavily rely on it. That's true.
I see. Thanks, Eardi. And yeah, Sam has a couple of questions, so I just allow him to talk. Sam, I think you can ask questions directly.
Yeah, I wanted to ask or explain maybe the notion of asymptotically I was thinking of, and see if that corresponded to what you were thinking as well, which was that if I had a certain error that I wanted to say get my computed mean within that distance to the actual manifold, then I could increase my sample size to a certain level, and so that my computed mean would lie within that error gap to the true manifold. And I was just wondering if that was sort of a notion of asymptotics that was the one you were also using.
Yeah, in terms of n going, do you mean n going to infinity here? So asymptotic as n goes to infinity?
Yeah, as n goes to
number
infinity.
of observations. Yeah. So yeah, the point I think I was trying to make, if I understood the question, sorry if I didn't, but is that there's only that much asymptotics can help. So asymptotic can help you getting so if I compute the population mean here, so for n that goes to infinity, that mean can be outside the surface, right? There might be that even for n that goes to infinity, that doesn't get close to the surface. Imagine this specific case, for instance, if I get an infinite number of observations, so I fill this surface with many, many observations according to the distribution within this surface. If I compute the mean as expected value, so the theoretical quantity, so basically n infinity, that would still lie outside this geometry. Does it make sense?
Yeah.
There's no concept of getting close to the surface for n that goes to infinity.
Yeah, I'm sorry. Maybe I was thinking of like locally.
Oh, that is okay. Locally. Yeah, I agree that locally, I mean, but if you're local, then it's arguable whether you need this machinery, because then you can basically fit a tangent space to your data. So the tangent space basically becomes a good fitting to your data already, right? So if you work locally. So then if I were to work locally, then I would probably throw away the entire Riemannian machinery. But yeah, locally things will work out.
Cool. I really appreciate your perspective. Thanks.
Okay, very nice. Other questions? I think people are very exciting for the Friday night. So yeah, so okay. So if no other questions, then thanks Eardi again.
Perfect.
And thanks everybody for participating.
Thank you.
Thanks, thank you.
Bye.
Bye.